Yeah, so good evening, ladies and gentlemen. As Colin has said, it's going to be a talk using lots of microtomography, and this time it's going to be a focus on amber, a truly amazing medium for preservation of ancient animals and plants. And I'm going to be talking about a Cretaceous coastal forest and a couple of stories about symbiosis, where you have interaction between animals preserved in the amber. And this is the first question. So, as Colin said, I'm a sedimentologist by trade. So, why is the turbidite guy talking about amber? And you can probably guess that it started off as a hobby, and that is the case. So, I noticed from about 2008 that you could buy amber online, and I saw some quite interesting pieces, and I started to buy them. And then I started to learn how to make images of these things because they involve high magnifications and very small depth of field. So focus stacking techniques. And I shared those images online and at the same time asked a couple of people, so George Poynar of Oregon State and Dave Grimaldi from the American Museum of Natural History in New York, saying, guys, if there's anything interesting here, let me know. So help me with the identifications, but anything interesting and you may borrow and work on. And both of them did borrow specimens. So this is one that was published by George Poynar in 2011. And we'll be looking at this very specimen a bit later, but in a microtomography data set. So we can get right in, move it wherever we want, look at all these little tiny nematodes that are escaping from the beetle over here. And George, of course, is famous for the idea of what if a biting fly sucked on dinosaur blood and dinosaur DNA could be extracted from the meal of that biting fly. So that's what he's to a large extent known for. Dave Grimaldi, both of these people are enormously prolific, but Dave Grimaldi has written a vast amount about inclusions and amber. And this is the one he borrowed, a crazy looking fly with wraparound eyes, which I then donated to the museum in New York. And he gave me and the family a day's run around the museum or wherever we wanted to go. In 2018, I published a paper with Andrew Ross of the National Museum of Scotland about Myanmar amber, and we'll be looking at this because it's really fascinating, and it's a kind of illustration of large inferences that can be made from careful observations on quite small specimens. And then last year, there's a publication, another one of my specimens on the earliest skink, a kind of lizard from the Cretaceous, about 100 million years old. And then also last year, there's a lovely team of entomologists and paleoentomologists who did me the honor of naming this creature. It's a girl. It's a pygmy grasshopper from the Eocene. So she's called Rusmithia gorochovai. And so from that, you can see that it's been quite a fruitful hobby. And the point I'd like to make here is that science isn't always very straight-faced. It's crucial that we test these hypotheses. Sometimes it's play. I wonder how you explain this. This looks really odd. Or I wonder what I could see if I can look at this in a different way, like with the microtomography. And that's what I'll be showing. So with these talks for the Yorkshire Geological Society, I like to make a link to Yorkshire, if possible. And we did that weekend before last with the Jurassic vampire squids. Now, amber can be found in Yorkshire, and these amber beads were brought here by Vikings and found in the Coppergate excavation. So you can go and see those either in the Yorkshire Museum or at the Jorvik Centre. And then I thought, wait a minute, on the coast, we have a set of beautiful plant beds from the Jurassic, from the Middle Jurassic. And that should be promising. And I have looked in the past, and I was reading a bit more about this in the last couple of days and discovered that there is globally a Jurassic amber gap. So there's amber found in the late Triassic, and there's lots of amber found in the Cretaceous, but not in the Jurassic. And this lady, Leila Seyfullah in Vienna, has been working on that problem. So really interesting. So anybody's on the coast looking at the Middle Jurassic, worth keeping your eyes peeled, because every now and again there's a small piece of amber that's found in Jordan or Thailand. So that would be super interesting. So my purpose, I'm going to be using microtomography to show how we can derive new insights from new techniques of looking at things through these three stories: Myanmar Cretaceous a coastal amber forest, a Dominican ant and a mealybug, a Dominican beetle and its nematode passengers. But it occurred to me that it's not just about this technology, and in that spirit of playful, curiosity-driven exploratory science, you never know what you're going to find, and sometimes it's valuable as a complement to other techniques, and we'll be seeing that as we go along. So here are those three stories. So amber ground borings in the coastal amber forests, and this is a rendered microtomography image that we'll be looking at in a bit more detail. And this is our mealybug in the jaws. You can see her mandibles there of a female gine with these pretty spectacular antennae that escape here. And then the final story will be about some hitchhiking, or phoretic as it's known, nematodes shown in pink there. So amber, what is it? So the other week I was reading Pliny's Natural History, which dates to 77 AD, and found it amazingly good. So he says it's a marrow discharged by trees belonging to the pine genus, a liquid at first, gradually hardened. And then he mentions the action of the sea, where fragments are carried to the shores. And that is how people collect amber on the beaches, washed up by waves in the Baltics. So surprising, really good. And you find amber from the Carboniferous onwards, but the abundance peaks are in the Cretaceous, the Eocene, and the Miocene. And as I mentioned, there's a Jurassic worldwide gap in the presence of amber. And this, what did he call it? Tree juice, something like that, Pliny. So it's produced by different tree groups. So in the earlier ambers by conifers, several different kinds, araucarians, or the southern pine, dawn redwood, umbrella pines for the Eocene. And then in the younger ambers, the angiosperms start to play a big part. And there's this tree, Hymenaea protera, that is the source of the amber in the Dominican Republic in Miocene. And this is a picture I took in Tanzania at the foot of Kilimanjaro. And you can see this tree weeping. So the amber is a defense mechanism. It's a sealing mechanism, making these stalactites. And you sometimes see that on trees here. And if you go up close, you'll very often see that there are little insects trapped inside it. So imaging. So I said it's not going to be all about microtomography. And I mentioned earlier on my first experiments, 2008 onwards, in making images. And the way you do it, so the way I used to do it is bring a camera up to the eyepiece of a microscope, focus the camera on infinity, and then focus with the microscope. So just move to each different level where you want something in focus, collect all the images, use stacking software to stack up and have an image that's in focus from front to back. And then later I realized actually you can have a machine to do that for you. And it's right here on the right-hand side of my desk. And I'm not going to turn the computer around to show you. So I've got a macro rail like this and a little box, and I tell it where to start and what increments and how many. So I might say I want 20 micron increments, I want 50 increments, and it goes snap, moves down, make sure everything stops shaking all the way down. And what you see here is a view from my Lightroom, and it's a new species of warrior termite currently in the review process. And each of these is focused at a slightly deeper depth than the one before it. And then when stacked up, you can have images like this with incredible details here of the limbs, which is important for recognizing what the species is. Now, microtomography. So X-ray imaging, X-radiography has been used for a long time, since 1895. But in recent decades, we've had this machine to play with. And this is Dr. Hijaz Kamal in Kuala Lumpur. And this is our machine. It's a Zeiss X-Radia X-ray microscope with a radiation hazard written in Malay warning over here. And this is looking through the window inside the machine. So we've got a rotating table in the middle. And we have an X-ray beam that emerges from this thing. And you choose filters depending on what you're showing. And then you have a sensor on this side. So as this thing rotates, catching frames, maybe 360 frames, what's recorded is where there are more dense things in the objects that you're rotating there. And the denser things cause a larger attenuation. And what the software does is reconcile all that information taken from the full 360 range of orientations, and it compiles it into a stack of slices, and often around 1/100th of a millimeter, sometimes less, sometimes more. And I'll mention the resolutions each time we come to a specimen. And I'm going to be showing you examples from in amber later. So I'm just going to show you this from last year. So this is something that I segmented, and it's the only stegodon tooth from Malaysia. And it's almost entirely in the rock. So what I've done here is, well, first of all, interpreted this as the enamel, and I've made the rock almost completely transparent. And then for the first time, we can see the complete geometry of this tooth. And the timing was very fortuitous because a research student had just finished his viva, and had misinterpreted every single component of this tooth. So just in time, we have the image and we can look at it internally and inspect it. And yeah, it can be very, very powerful. All right, story number one, the coastal amber forest. And there are lessons in here about caution in our interpretations that are quite spectacular, actually. And Myanmar is the place where this amber that I'll be discussing comes from. And it's a spectacularly beautiful country, and a very troubled country as well, of course. And I've only been there in 2019, in a brief gap between political troubles and the onset of COVID. This is Shwedagon, this spectacular pagoda in Yangon. And these are two of about 1,000 temples. They're about 1,000 years old, plus or minus 100 or 200 years, at a bend in the Irrawaddy, Bagam. So it is a spectacular place to go. But I didn't get up to the area where the amber is mined. So here's the general area. There's Kuala Lumpur over here. And if we come over to the detail here, here's Shwedagon, where that pagoda was. Here's Bagan of the 1,000 temples. And then Noyabum and Mitkina, where there's famous large amber markets. They're right up in the north, a very troubled area in the Hukong Valley. And I never made it up there. So that's today. And then back 100 million years ago, this is where that area was. So here's the equator. So it's a low latitude island arc, Tethys opening up over there. So the weekend before last that talk, we were looking in this area, up in the narrow bit of Tethys. But here we're along that northern margin. And this set of amber fossils is outrageously diverse. So Andrew Ross up in Edinburgh, he collates all of the new papers, all the new species descriptions. So you can see on this axis, 1999 through to 2023. And in blue, we have all taxa that have been described. And you can see it takes off around 2016. And it slowed a little bit, 2022, 2023. That's 2,805 species described to date. Plants, animals, invertebrates, vertebrates. And as you can also see, a large proportion of them are hexapods, insects. And he keeps that up to date. So everybody, when a paper is published, it's sent to Andrew and he updates that database. Now, this is just a bit of fun. So I'm going to show you an animal from the modern. And each of those modern pictures is from Northwest Borneo, from a forest. It's a slightly lower latitude than Myanmar was. But this Myanmar amber is an astonishing, rich sample of a Cretaceous rainforest biota. And you see many of the same groups of animals today as you go walking in Northwest Borneo. So this is a a whip scorpion, and incredibly rare, this thing over here, Mesotheliphonas. But you can see the whip on its tail over here. And there are other arachnids. There's one called the whip spider, which is a couple of whips up at the front, basically. And very large centipede over here. And this one is an amber, 6 centimeters long. It's quite a big specimen. And here's a tarantula. And I published this specimen on the right, not because it's an amazing looking spider, but because you can see this structure here cutting through it. And that's what we'll come back to later. And you look a bit closer, look at, so he had these 4 eyes with a little dose of pyrite in the middle. Paul Selden wants to work on that, because there's another thing there, which is it's got its color patterns preserved. It's got these white stripes. So what we've got here, one of these harvestman spiders, this is on the Kinabatangan in Sabah, northwest Borneo. And yeah, here we've got a damselfly. So you can see the big eyes in both of them. This one's wings are spread out on both sides. Quite similar. And also mollusks, like this snail. This is from the Danum Valley in Sabah, and in the amber. So here's a similar looking snail, and with its operculum still in place over here. And these are very dramatic to find in the modern forest. And here's a bug with a large rostrum in the Cretaceous amber. And there are vertebrates as well. So there are a couple of frogs, there are some snakes. This is a skink in the modern, and this is a skink in the ancient. So this is something which should be out this year. And I donated this specimen to the National Museum of Scotland. All right, so many specimens of amber from Myanmar show this feature that's on the slide right now. Because so you can see there are these circular sections, and there's this expanding geometry as you go from the outside of the amber towards the middle. And what are they? So I didn't know what they were, but this is a brilliant paper, Grimaldi et al. 2002, which is a spectacular amount of work. It's a very comprehensive overview of the biota, the Myanmar amber biota. And he also saw many examples of this kind of thing. And he thought that what they were fungal sporocarps, fungal fruit bodies. And on the right here, we've got an explanation. So here is a depression in the bark of the tree, and these are the fruit bodies of the fungi. And then that fills with resin over here, and then later this pulls away from the tree and is found separate with all of these structures around the outside. But he's good because he says possible sequence, so he's a little bit cautious, rightly so. And then we've got Poyner and Brown, 2003, which strangely doesn't cite the Grimaldi paper, but he now gives it a name, Paleoclavaria burmitis, and gives the name to the new family, Paleoclavariaceae, however you're supposed to pronounce that, I don't know, and very confidently describes them as fruit bodies and hyphae. So here again is an image similar to the one I showed before. Zooming in, it infers that he's looking at spores and something called cystidia, these little lobes, and presented with a lot of confidence. And in the spirit of play, I knew that this would make a nice image. So I took that specimen I showed you earlier and stuck it in the X-ray microscope and got images like these. And this thing here, this is a leaf of a dawn redwood. And you can see it just chops all the way through. So there's a cross-cutting relationship. And yeah, these are just two other views. So you can see a narrow neck, and each of them widens out like this. And so I actually wrote to Dave Grimaldi and said, so these fungal sporocarps, I'm seeing cross-cutting relationships, and also they're full of sediment. Can you see here? Which is why that image was so nice, because we've got a really big density contrast between the sediment-filled structures and the amber. And he said, you have just found my most embarrassing scientific mistake. And I offered, I said, shall we write it up together? And he wasn't interested. So I wrote it up and joined with Andrew Ross. So here you can see coarse lower sand. So this pebble of amber has ended up in a sedimentary context. And these depressions, these holes have then been filled mostly with sediment, sometimes with calcite, like you see there. So that's 37 millimeters across this specimen here. And in these 3D images, you can see 1, 2, 3. That's these over here, 1, 2, 3. So you can see these have been chopped across. And then another observation. So far we've got cross-cutting relationships, we've got sediment fill, and in another specimen, I found the actual bivalves still inside their crypts, domicnia it's called, their dwelling structures. And this one is amazing because you can just about see a crisscross pattern. And that's the mark that it makes when it rasps, when it's actually boring into something hard. So we've got those 3 observations. And yeah, it's clear by now. De Grimaldi knew this as well. So they actually are boring bivalves, piddox, if you like. But then there was another observation, and it's this. So here's a plant, and here it's not cut across by that bivalve. This bivalve is just floating in the amber, and it deflects the plant. And this one over here, instead of occupying that nice club-shaped structure, it's got a really irregular structure that comes up to its end, filled with sediment here. And this is on the right is that left-hand image in the micro CT image. So I've said here, regularly shaped sediment-filled structure on the outside of the amber, and various details of what these piddox look like. And here's another view of that irregular structure. So. And the other thing to see here is that the shells are broken. So there's been movement and the things have actually broken. And I didn't know what to make of that, apart from the bivalves had got into the amber and they were floating in the amber and they weren't in the tidy borings that we're used to. Pteridolites, you'd call it in the modern, or gastrochenolites, if something had bored into something hard. And then I read this paper by Schmidt and Dilcher, and this is a paper looking at the modern, this is in Florida, looking at amber, looking at resin as it reaches water. And it takes a long time to harden in this case. And for example, here you can see there are inclusions that have just swum through the very thin skin and have become trapped inside. So my argument was, okay, so wherever this amber was produced, it was in a sufficiently high salinity environment for the little spat of the piddox to land on the amber before it was hardened. So you can imagine them starting to bore away and finding themselves inside a sticky mess, basically, and not boring through something hard. And a bit nerve-wracking because I couldn't think of another explanation, and that's the one that was published. But then if you look at these 2 very famous authors who have entirely misidentified the most common structure in the Myanmar amber, it makes you think, doesn't it? So where have I messed up like that? And where have I stopped at the first interpretation because I couldn't think of another one? And it may be, it's shortage of imagination or shortage of knowledge, but it just does emphasize the need for caution. So just after that paper was published, more evidence started coming in. And there was a tapeworm, and there were marine gastropods. And then the most spectacular one is this. It's an ammonite. And there's another interesting point about the bivalves boring into soft amber, because it has been suggested that the amber could be a lot older than the sediment that encloses it. And the argument would be that amber pebbles have been moving around for a long time, bored, and then much later buried. But what those observations about the little tiny ones boring into the soft resin tells you that those bivalves are very close in age to the amber, and also that the amber forest, the trees that were supplying that resin, tree juice as Pliny called it, were in a coastal setting. So tiny little observations, but a lot to be inferred from that. And this ammonite, so it's a juvenile, it hasn't got any soft parts, it's a little bit broken. So it's found its way into amber, presumably also in a coastal setting. And its age is interesting. It's Upper Albion through the Cenomanian. So those are the units that straddle 100 million years, more or less. So that confirms the age of the amber. And yeah, you wouldn't be too precise, but about 100 million years is what you think. So here is northwest Borneo and Sulawesi, Indonesia over there. And this is the Baram Delta, this spectacularly sinuous river channel that comes out here in a big plume in the front. And this is Brunei Bay. So this is an area that I visited pretty much every month for several years, or at least every other month. And what you can already see from a distance is there's forest right up to the coast over here. Less so on the Malaysian side, where they've cut down most of the trees, but in Brunei, it's virgin forest, mostly pretty much up to the water. So this is the kind of thing that I had in mind. So that was the first story, and it's a bit like the vampire squid story from the weekend before last, where we make observations on a small scale that are very relevant for the larger scale. And then once those ideas are out there, people can come and disprove it, of course, but they can also go out and find more evidence to strengthen or weaken. And now the next part of the talk, I'm going to be focusing on the remarkable images that you find in amber of interactions between animals, different kinds of symbiosis, animals and plants, of course, because here is a bee with its legs utterly laden with pollen. So that's one kind of mutualistic symbiosis. And this is going to be the next story. This is a mealybug with an ant, and that will be the next story, which is the hitchhiking parasitic nematodes. But this one is really fun. This is from the Eocene, and it's a fly, and there's something down here hanging onto a leg. And when you look closer, it's a little pseudoscorpion. And I should have put a photo in of a pseudoscorpion not hanging onto something, because they have these large arms and very, very big fat pincers. But this is how they get around. They find a bit of transport in the form of a fly, and they hang onto the leg and get taken to somewhere that they like, and then they get off. All right, so symbiosis stories in Dominican amber. So this stuff is a lot younger. So the amber from Myanmar that we've been talking about is around 100 million years old. And this Dominican amber is probably 15 to 20 million years old. Some of it may be older. It's quite hard, actually, in general, dating amber. And so this was Myanmar. I'm just going to take you on a little tour below the Himalayas. And up north of us would be the Eocene amber of the Baltic. And this is where we're landing. And La Cumbre is one of the areas where there was a lot of amber mining. And again, it's tropical rainforest biota that's sampled, but packed with beautiful fossils. So first story here is trophobiosis. So it's a symbiotic relationship between organisms where one of them obtains food from the other one. And the one that provides the food is called the trophobiont. And the food in this case is honeydew. And this is a specimen that I bought 2008 for not very much, and showed it online. And I think it was George Poynter said, I think, because I thought, is this some kind of predator-prey thing? The ant's carrying something there, isn't it? And he said, no, I think what you've got is something very, very rare indeed. And directed me towards some quite recent literature about ants and mealybugs. And this is an important paper, Johnson et al. 2001. And this is from French Guiana. So can you see, here's the ant, antennae here, mandibles. And what is it carrying? It's carrying this plump little mealybug very firmly with the mandibles. And here's a drawing here. This is in South Africa. You even get them in Europe. So you find them in Italy, and you find a few species in Malaysia. And it's a kind of ant that is subterranean. And that's why you very rarely see them, apart from when there is a mating flight and the females with their wings go flying off to establish a new colony somewhere else. And what they do is they take with them the mealybug to start a new colony of mealybugs that they can then harvest the honeydew from. And this is a table, but these are all species of Acropyga down here. And what I wanted to show is what an amazing number excuse me, of relationships there are between Acropyga ants of different species and the different mealybugs from all over the place. And this is the first paper that mentioned discovering that phenomenon in amber. So not very old amber, 20 million-ish Dominican amber. And these are the images in that paper. And I've mentioned this mealybug here, Electromyrmecoccus inclusus, because that's what I'm going to show you shortly. And so my example is just the second example of this, so it's really quite rare. So how rare? Here's a quote from that paper. 3 pieces of Dominican amber, each containing an Acropyga gyne, so that's a winged female ant, with a pseudococcid, were recovered from approximately 30,000 small pieces of Dominican amber screened by D. Grimaldi, so 1 in 10,000. And it's not that surprising because most of the time they're under the ground. These are not ants that will very commonly walk into amber if you're living above ground, but it's when they head off on one of these mating flights that they get trapped. And these are images of my example now, and we can probably contrast them with the 2001 images. But here's a general view, and here is that mealybug with the wings, and this is what's called the gaster and the mesosoma. It's really quite twisted in the rock. And then this in the middle is my interpretation in a 3D microtomography data set. Here's the scale of 1 millimeter. And it's really, really hard. So the weekend before last, we had an object that was pyritized in a mudstone matrix, which was not so difficult to segment at all. But here, the density contrast is very small. So that image took a lot of work. And you can see actually that it's not as good in some respects as the photo. So these little anal lobes of the mealybug, the CT that are there, these little hairs on the end, they're not captured in the data set. And this data set has a resolution of about 10 microns, so 100th of a millimeter. And this is the photo stacking. It's the kind of image that you can get with the photo stack. So look, I've not made it sharp all the way back. I've just gone from the head, but it's quite a beautiful image of the mealybug back to the ants of this Acropyga ant. And so what's the story? And this is one of the things when you're being curiosity driven and just taking a look. So you think, I wonder what I'll see if I look at that in 3D. And in this case, you end up with the same story. It's very clearly one of these female ants that's carrying her mealybug, starting a new colony. But there's one thing that's quite special about the 3D view, which is that you can view it from any angle. And with the amber, you can't. The amber is generally flat, especially when it's been polished to come quite close to the inclusion. But you can be very precise with the measurements. So what I've done here is, in green, I've shown, there's a guy, La Poja, in 2005, who measured a number of these ants to 3 decimal places, which surprised me, because it's given that it's just 2D views and plenty of potential for parallax. And these big blue circles here, this is my specimen that I've just been showing you. And the purple ones here, that's a male, that's one male ant that he had to look at. But you can make really quite precise measurements, because you go to one end of the thing you want to measure, put a little dot, go to the other end of the thing you measure, put a little dot, and the measurements are good. And this I've done in the last couple of days. So that first image that I showed you was done with software called Drishti, meaning light in Sanskrit, which is written by AJ Limai of the Australian National University. And I thought, and it was really hard work, I thought, let me try with a different piece of software and see if I can do it quicker and better. So this is with 3D Slicer, that's very widely used medical software. And we'll just spin it around so you can see how twisted that ant is for a start, and how very difficult it would be to make good measurements. But it was hard. The blue are bubbles, and those were segmented very easily. I'd grow from seed. I said, this is a bubble and this is the amber, and the program got it well. But the ants and the mealybug, it's composed of 2 different thresholded segmentations with masks, and also some hand painting. And you can go much further than this, but it would take a lot of time, and there's not really the purpose to justify it. So interesting. Each piece of software has its pluses and minuses, it seems to me. Well, the two that I've tried, I've only tried two. And now we're on to hitchhikers. So we're still in Dominican amber. And Foracy, you see described there, special kind of commensal relationship in which one organism attaches to another for a limited time period to enhance dispersal of the forant from the birth habitat, often resulting in colonization of new and potentially better habitat. And that was also the story for the pseudoscorpion that we looked at earlier. So these are George's pictures from 2011, and they're pretty good. So here you can see that there's an adult nematode just on between the two elytra of this beetle, a platypodid beetle, and these are all the little ones, the dower as they call. This one's worth noticing because we'll find it later in the 3D data set. And here he's gone close up to see one of these nematodes with its intestine down the middle, and that scale bar there is 31 microns. So that is about a third of the width of a typical human hair. So the whole nematode there is the length of two human hairs laid side to side. So if I remember right, a human hair is typically 60 to 120 microns, something like that across. So very, very tiny. And it's quite a dramatic image, but at a really tiny scale. So here is a new photo of that platypodid beetle and all here's that kinky, the wriggly one, all the little baby nematodes. And you can see that it's quite discolored, and the amber is a bit murky in places. It's also a very flat piece of amber. So we've got basically this side or the underside we can look at, and the image is blocked by cloudy parts of the amber. And once again, in an X-ray microtomography data set, I've gone in and interpreted the thing. So the platypodid I've used thresholding for. But again, the density contrasts are very small. So each of these purple things is a nematode that we've seen in the photograph. But I've had to do those by hand. Often in a tea room in Kuala Lumpur, with people walking past and looking over my shoulder and they're saying, what are you doing? So doing this, you start to see that there are many more nematodes than we ever realized back in 2011. About 35 of them I count. And just again, a couple of days ago, I've just been playing with the transparency. So here's the beetle, here its eyes. And these are two bubbles that are trapped under the right elytron of the beetle. And this is the mass of nematodes. So I took measurements, and you end up saying, one of the nematodes managed to reach 800 microns away from the beetle. And you look at the picture and you think it's very, very dramatic because they've hit the resin, and the little nematodes go swimming for their lives. Swim, little nematodes, swim. None of them made it. So all on a scale of less than a millimeter. All right, have I been really fast? I have. It's probably okay because there's probably going to be lots of questions. These were the three stories that I brought to you. So the first one was looking at these club-shaped structures and taking them from being interpreted as fungal fruit bodies, complete with lengthy formal names and formal family names, through to them being bivalves, so boring bivalves, piddox, and looking at more specimens, realizing that some of them were colonized before the amber was hard. So they're contemporaneous with the production of the amber, of the resin from the trees. So that was that story. And then here what have we got? We've basically got much better images, both photographic and in 3D models. And we have a much better set of measurements. But I'm not really sure what to do with this one. So I put it on a preprint server for ant specialists or mealybug specialists that will find the information useful. And this one I've also not done anything with yet. And sometimes it's like that. So you go exploring. You think, what might I see if I look at that in 3D? And sometimes the story doesn't change very much, but you can greatly enhance the images that you've made. So, conclusions. Hopefully you'll agree, microcomputed tomography is a very powerful tool for visualization and analysis. So you can see in 3D, you can pick any viewing angle and you can measure anything you want. And what we saw the weekend before last was you can also scale up and export and print objects that you're interested in. And this was the story of the pholads, cross-cutting relationships, sediment fill, the fossils actually within the borings, and that overturned that. So this one actually overturned the previous interpretation. And the next observation, the landing on resin that wasn't fully hard, tells us something about where the forest was and the age of the resin relative to the borings. And that was really satisfying. The paper's very, very well cited because of the timing, I think, because after that, new evidence was found that supported it. There was also another paper, actually, that found these boring piddocks tolerant of low salinities in the lower reaches of rivers in the present, in the modern area. And I reviewed that and said, yeah, this weakens my story, but yeah, recommend it to be published with some changes. And there's a phrase in that paper now, which is my phrase in the review now plonked in it and recited. So good, all good, but funny. So we can make accurate measurements, and that was the point in the ant mealybug story. And that selection of unlimited viewing angles is really important because very often you find that you're very restricted in what you can see, and you miss a lot. Or there's a potential for not seeing a good proportion of what's actually there in the specimen. And that's the final conclusion. Sometimes really big deductions can be made from little specimens. And obviously, I encourage, especially youngsters, to play, just follow your curiosity and see if it yields something new and different, or if it just yields satisfying 3D images and better measurements. But yeah, exploratory science, just try it and see, have a play, see what you see. And that was quite fast, which I didn't think it would be. And I think that's because you're all silent and I don't interact with you. But thank you very much for your attention. And now, let's see.
Rue, do you want me to ask the questions on behalf of the attendees? Okay. I can do that. I can actually see them coming in now.
So that'd be really good. I would have loads of questions.
Thank you very much for the presentation. Really spectacular imagery, really. It's a shame we can't get a virtual applause, but first, I think it's more of a point of interest from Nick Riley, saying Carboniferous records of amber come from coal mines in Kentucky, USA. These are disputed as being Carboniferous, but may have come from pit props. So I don't know whether that's, because you said they go back as far as Carboniferous, but whether that really is.
Because that's really interesting. There's something called resinites that I've seen from UK coal mines, which looked very suspicious to be well. Is it amber? Doesn't look like it because they're big blocks. So I believe what he just said there. But the other thing is that good amber is found in Idaho in the Carboniferous, and it's quite remarkable because its composition is very much like the Dominican Republic amber, which is Miocene. And there's a nice short article by Dave Grimaldi in Science 2008, which discusses that and then emphasizes the caution that you have to exercise in trying to infer what type of tree produced the resin from its composition, because the Dominican amber is produced by an angiosperm, Hymenaea protera, and that certainly wasn't the case for the Carboniferous amber. So there's quite a big community out there. I see. I only skim, but worrying about what controls when amber's abundant, when it's not there at all. And there are lots of correlations with climate warming, potentially abundant forest fires. Resin being a tree's healing mechanism, so defense, sealing, and healing mechanism. And how do those correlate with other factors? So that lady Layla Seafella that I mentioned, I've seen that she's got quite a sequence of papers which I haven't read on all of those. And there was a massive paper by Delclos et al. And I don't know, it looks like 50 authors talking about the Cretaceous resinous interval and what caused it. Why is it there? Why did that happen in the Cretaceous? So interesting stuff. Yeah, thank
Uh,
you.
Next question. This is from Shell Simpkin, and it says, "Does amber taste good or provide anything to these bugs?" So this is where we ask you, did you actually try eating it?
This time I didn't. Yeah, for those that weren't there weekend before last, I did a dissection of a modern squid and photographed it, and then ate it, then cooked it and ate it with a really nice glass of wine. But no, I didn't do that with the amber. um, but she was asking if anything eats it, was she?
Yeah.
But
So
yes,
is there something beneficial about the amber that makes them attracted to it, or is it just unfortunate being caught in it?
unfortunate. Yes, yes. So either you fly into it, or your feet get stuck in it, and then another flow goes over the top. And I've got some beautiful specimens, which, given the amount of time, I could have actually filled some more of an initial stalactite, like the Tanzanian example I showed, and with a wasp on it, and then another stalactite around it, just enclosing that wasp. So one got stuck, and then the next flow wrapped around the poor old insect, leaving it there for posterity.
Uh, I asked for some more questions to come in, but whilst we're waiting for some more, I just think that with the Myanmar example where you showed the ammonite that had been trapped,
Yes,
why was there no soft body parts to it? Had it died and got caught up in this? I would have thought it would have preserved, if he'd been living, it would have preserved some of the soft parts.
that's right. So that's an important observation. It would have been triply spectacular if it had
Mm.
soft parts, because nobody knows really what the soft parts are for ammonites. There's some indication that they've got a couple of longer arms like my squid, um, but it tells you it was a dead shell, a little bit abraded. There were marine gastropods with it, so that has been washed on to a beach. There were also little grains of carbonate sand there, or shelly sand, included with it, along with quite a few terrestrial inclusions: cockroach, flies, spider, beetles. So it was a real mixed thing. So that's presumably a few marine fossils on a beach that are washed into an environment where the resin can fall and enclose them. And it's also at the same time trapped the proper terrestrial organisms that were living on the tree or close to the tree. Really fascinating specimen. And I was up visiting Andrew Ross depositing the big lizard, and he said, "We've got an ammonite." And you always think, "Hang on, is this a wind up?" But no, it was actually real. Ammonite in amber. Yeah, I'm not going to get trilobite in amber, but an ammonite in amber. Yeah, true and incredible.
And again, there's no more questions in, but I've asked about the nematodes as to, I find it quite amazing considering the viscosity of the amber. You think they're actually trying to swim away from the body of the insect.
Yes, because they were all hiding under the wing covers, the elytra, the tough forewings of a beetle, and that's where they all were. And the adults were still there under those wing covers, but the little ones had scrammed in all directions, hadn't they? So they'd tried, and yeah, it would have been liquid, of course, and hardens over time. But it makes me laugh the scale of it, because it's such a dramatic story of attempted escape, but no, didn't happen.
I see no more questions. So at which point, again, thank you very much, Rue, for this really fascinating talk. Spectacular images, really. And hopefully we'll see some more developments in the future with this.
Yes, yeah, it's a fascinating subject, isn't it? The material itself attracts, and the preservation of what's trapped in it
Yeah.
is astonishing. So yeah, you look at something which is 100 million years old, but it looks as if it was alive this morning, which is incredible. So very
And
entertaining.
you've got more collection to work on, have you, that you bought?
Yes, but the two machines that I use are not working. They're broken now. But I've got so many other data sets to work through that I don't need to get more.
Brilliant. That was really fascinating. Thank you very much.
Thank
And
you.
for everybody, if you can make it along to the Leeds meeting on the 1st of March, you're more than welcome. Okay. Thank you very much.
Uh, I shall stop.